Hey AP Bio, so here are the answers to the homework questions for mitosis, the cell cycle, and meiosis. So the first question here says, explain how cell division in prokaryotes differs from cell division in eukaryotes. Prokaryotes, they divide by binary fission, okay, which means they just simply take their single chromosome, they replicate it, and then they just split into two, and they just divide the cell evenly. So there's nothing special that they need to do because they only have one chromosome. Therefore, there's nothing that they have to organize and arrange and move around to make sure that they equally can distribute the DNA. Eukaryotes have multiple chromosomes, and because of that, eukaryotic cells must use mitosis to organize and divide their DNA. Um, the chromosomes need to be correctly organized. They need to be moved to the middle of the cell so that you can properly separate them and ensure that you have equal amounts of DNA going into each cell. Cytokinesis then occurs after that to split the cells into two. Okay. Again, nice short question, quick and easy to answer. Okay, let's move on to the next one. Describe the structure of eukaryotic chromosomes. Okay, so eukaryotic chromosomes basically consist of highly coiled DNA, right, which is bound to proteins called histones. Those histones, they're going to help with that supercoiling that goes on. And that allows us to condense our DNA into chromatin, into these very distinct structures eventually for replication and for mitosis called chromosomes. Um, but our DNA is highly coiled and it's highly condensed simply as a matter of practicalities. We could fit it all into one cell. Okay. All right, let's move along. Human cells, they divide every day for normal growth. And so this tells you this whole thing about this epidermal growth factor cell. So when you get cut, damaged skin cells secrete this protein called EGF, which will signal cells to begin going through cell division. So the three major stages of interphase and describe the sequence in which they occur. So we have G1, GAP1, S synthesis, and G2, GAP2, and they occur in that order: G1, S, G2. The second question they asked you was to explain how a mutation outside the coding region of a gene could lead to overproduction of the protein in a skin cell. Well, if you have a mutation outside the coding region, that could affect a signaling molecule. So there might be a molecule that activates the EGF and tells the cells to grow. And if that mutates, then the cells will grow all the time and become cancerous. Okay. All right, move on to the next one. P53 protein. Here we go. Okay, so they're showing you this diagram of P53, and it's a transcription factor that regulates a cell's progression through the cell cycle after DNA is damaged by UV radiation. And so they show you this pathway. So you can see you have damage that triggers kinases, which activate P53. And then P53 then activates this thing called a cyclin-dependent kinase, or CDK inhibitor. Okay, so it's going to inhibit something. Now, if you remember from the notes, cyclins control cell division. So if you inhibit a cyclin, you're going to inhibit cell division. So for letter A skin cell completes one round of the cell cycle. Describe the products. Well, the products of one round of the cell cycle are two daughter cells that are identical to each other and to the parent cell. Letter B, they want you to explain how P53 regulates the cell cycle in the presence of damaged DNA. So in the presence of damaged DNA, it's going to activate a CDK inhibitor, which stops the cell from dividing. It stops the cell cycle. It prevents the cell from going into mitosis. Okay. And for letter C, explain how a mutation in P53 leads to an increased risk of cancer. Well, damaged DNA is going to get copied and inherited by the daughter cells. So if any of that DNA that's damaged happens to contain regulatory genes that control cell growth, the cell's going to grow out of control. Okay, let's move on to the next one. Okay, we have a student who collected samples of cells from a woody plant during the spring, and they produced these drawings below. So sample 1 was from the woody stem, sample 2 was from the root tip, and you can see that there are cells in different stages of mitosis, which are labeled A, B, C, D, E in sample 2. And so they made these drawings, and we can see that there's a lot of mitosis going on in a root tip. So they want you to describe the role of mitosis in the growth of a plant. Well, mitosis is used so that the cells can divide, and it's a way for more cells to be produced in plant growth. Letter B asks you to explain why there are more cells observed in mitosis in the root tip, and that's because the root tip is an area of growth. If you see a lot of mitosis going on in an area, that means that it's growing, that it's actively growing, and there's an increase in the number of cells in that area. The woody stem doesn't exhibit much growth, and we know that because in this woody stem there are only a couple of cells that appear to be in mitosis. The rest of these cells are in interphase. Okay. Letter C says refine this model in the space provided for your response by using the letters to represent the sequential steps of mitosis. So if you look at this E, D, B, C, those would be the sequence of steps. And if you look at this, E would be prophase, metaphase is D, anaphase, you can see them moving apart. And then the last one is C, it's telophase, where you're starting to form those two new cells, and you can just start to see that that cell plate is forming in there, splitting the cells into two. And then the last question says, explain how the arrangement of the cellular components during the step of mitosis portrayed by cell D in sample 2 facilitates the proper distribution of chromosomes to the two daughter cells. Well, look at D, chromosomes are in the middle. What's going to happen? Those chromosomes are going to get pulled apart. So those chromosomes are aligned in the middle of the cell so they can be evenly split and distributed to the new cells. Okay, those are your correct answers. Let's go to 6. This one I explained a little earlier. So this MDM2 normally inhibits P53, and it prevents the production of active tetramers, which shut down the cell cycle. So if there's less MDM2, right, if MDM2 is not there, that means P53 does not get inhibited. So P53 will be active, and it will in turn inhibit the cell cycle, which reduces the process of mitosis in the cells that form the red blood cells, which are those multipotent stem cells. Okay, so with less MDM2, P53 becomes activated, and then it'll inhibit the cell cycle in those cells that form the red cells. And the letter B, it says predict how a loss of function mutation in the P53 gene is most likely to affect the cell cycle in response to DNA damage. Justify it. The cell cycle will continue without any response to DNA damage. Therefore, mutations will be passed on to future generations. Okay, so again, without MDM working completely, the P53 gene will most likely not function properly, and there'll be no response to DNA damage. Therefore, you'll have all these mutations being passed on to future generations. Okay. Okay, let's move on to the next one. I had explained this one earlier in a previous video, so I'm going to just jump right to the answers. Capsaicin, they want to know what are the characteristics of capsaicin that allow it to enter the cell, and describe what prevents NFAT2 from inhibiting ATF3 expression without calcineurin. So capsaicin, to get into the cell and enter the cell before binding to a transmembrane domain, it has to be small and nonpolar so they can easily pass through the plasma membrane and get to that transmembrane domain. Okay. And in the absence of calcineurin, that NFAT2 cannot be dephosphorylated. So without calcineurin, you don't take the phosphate off, which means you don't activate it. So NFAT2 remains inactive, and so as a result, you will not be able to affect AF3 inhibition. It says here letter B. The protein PGOG is known to indirectly interfere with interactions between cyclins and cyclin-dependent kinases. Explain why this prevents DNA replication from happening in the cell. Well, when you block a cyclin, the signals for replication are inhibited. Cyclins signal DNA replication to occur. So if you inhibit cyclins, you're going to inhibit cell division. Okay. For letter C, they discover a mutation that results in constant expression of ATF3. So ATF3, regardless of NFAT2, gets expressed all the time. That, in turn, they want to know, predict the effect of this mutation on the cell cycle of cells treated with capsaicin. So if you treat the cells with capsaicin and AFT3 works regardless of the presence of NFAT2. Basically, it's not going to have any effect. Right? So what's going to happen is constant expression of ATF3 is going to result in unregulated mitosis. That could lead to cancer. Okay? So cells that are treated with capsaicin won't respond. You'll get constant expression. Okay? Letter D says researchers claim that capsaicin could be used as a potential treatment for melanoma, a type of skin cancer. And we know this because capsaicin causes inhibition of cell division. Cancer is uncontrolled cell division, so this would stop additional cell division from occurring because capsaicin would essentially inhibit the pathway that activates PGOG. Okay. All right, let's go to 8. Outline the following events that occur during prophase 1. So synapsis, that's when the homologous chromosomes, they actually form pairs. Tetrad formation is the actual formation of those homologous pairs, and it consists of 4 chromatids. Crossing over is simply the exchange of sections of chromosomes during synapsis. That leads to increased genetic variation. Okay, we'll go to number 9. How does meiosis provide for greater genetic diversity? Okay, it does this through the sorting and the recombination of chromosomes, right? Those chromosomes get separated from each other. We create new combinations that didn't exist, and also through crossing over. In meiosis, once the cell divides twice and produces 4 haploid cells with unique combinations of chromosomes, you're going to see variation that you didn't see before. Okay. And for number 11, they want you to describe 2 events that are common to both mitosis and meiosis that ensure the resulting daughter cells inherit the appropriate number of chromosomes. So for letter A, DNA replication, that's common. You want to make sure you have double the amount of DNA so they each get the same amount of DNA. Okay. The formation of the spindle fibers is going to align and assort the chromosomes. That's going to ensure that those chromosomes are lined up and that they get properly separated and assorted. That happens in metaphase and anaphase. And then we also have cytokinesis, which permits cells to split after the DNA has been evenly distributed. Okay. Letter B tells us that the genetic composition of daughter cells produced by mitosis differ from that of daughter cells produced by meiosis. Describe 2 features of the cell division processes that lead to these differences. So the first one in meiosis is crossing over, which leads to increased diversity. The other thing is that in meiosis, DNA replication only occurs once. So you're going to wind up producing cells that have half the normal number of chromosomes. So the result of this is cells that are not the same as what they were from the initial parent cells, because you have now crossing over, which is creating more diversity and mixing of alleles. And the second thing is you have half the amount of DNA. So having half the amount of DNA, of course, you're not going to have the same genes that those original cells had. Okay, and the last one, number 11. Describe 3 differences between mitosis and meiosis. So these differences, cell division occurs twice in meiosis, once in mitosis. Crossovers happen in meiosis, but not mitosis. Homologous pairs, they'll form tetrads in meiosis, but you don't get tetrads in mitosis. Okay. A couple other things you can add. Mitosis produces 2 cells that are identical. Meiosis produces 4 cells that are not identical to each other or the original cell. 2 differences between gametogenesis in males and females. Okay, so in males, you're producing 4 gametes. In females, you're only producing 1 viable gamete. Okay. 2 examples of meiotic errors and their effect on offspring. This could be anything from Down syndrome, trisomy 21, Klinefelter syndrome, Turner syndrome, supermale, trisomy 18, trisomy 13. So you can have a couple of these different types of trisomies. Those are examples. Okay. And that's it. Those are your answers for the homework. So I hope that helped. Again, please reach out if you have any questions about any of the homework assignments, and I'll see you guys in school. Have a great night. Take care, guys. Bye.